Great. Thank you for joining us. Yeah. Oh, I project. Thank you for joining us for the UW Data Science Seminar. Today we have a speaker from our AI Accelerator program that we ran in the fall. If you're not familiar with the eScience Institute AI Accelerator, I encourage you to go to our website. It is a cost-free way for you to get support to get through technical or intellectual barriers that are prohibiting you from making exciting progress on an AI or data science related project. Um, Talking about using AI models to predict cross-family ligand binding. Today is Ike Keku. Ike is a bioengineering PhD student. He works in the lab of Dr. Princess Omukade, where he studies systems biology, data science, and protein-protein interactions. I had the privilege of working with him last quarter, which is why I'm up here today. He completed his undergraduate work in biomedical engineering at UNC Chapel Hill, and I appreciate your attention to him today. Thank you, Ike.
Thank you so much, Dave, for introducing me, and then also Nicoletta for having me. Again, I'll be presenting about using AI models to predict cross-family ligand binding to VEGFR. And then I'll also be talking about my project with the eScience, Data Science, and AI Accelerator program. But before I wanted to do that, I wanted to start off with some background information, and then, of course, then transfer into the work that I did in the autumn quarter. So before we dive into signaling, I wanted to ground ourselves in what proteins actually do. Proteins are the main workers of the cell. They can act as receptors on the cell surface, enzymes that carry out reactions, structural components, transporters that move things around, and messengers that pass along information. And when we build models, we're often tracking how often these proteins interact, change states, or move between compartments in response to signals. So signaling is basically how cells talk and listen. The signal might be a growth factor, a hormone, or a stress signal from the environment. That signal binds to a receptor and triggers a cascade of events inside the cell. These cascades control things like cell division, survival, movement, and differentiation. When we model signaling, we're asking, given a certain signal, how does the cell respond over time? And what happens if we change parameters? Signals are detected by receptors, and there are multiple receptor types. These include intracellular receptors, which regulate the intracellular processes, and then also ion channel receptors. The schematics for this are on the screen, but just to give a very brief overview of what they do, intracellular receptors, they bind molecules that can cross the cell membrane, and then ion channel receptors open and close so that ions flow inside and outside the cell. Then there are also G-coupled protein receptors, which aren't shown on the slide, but are another type of receptor. Then there are enzyme link receptors. And these receptors, systolic domains, have enzyme activity or associate with the enzyme. So in the diagram on the right, you see that they have a ligand binding domain, a transmembrane domain, and then also an intracellular active enzyme domain. Now, these include serine 309 kinase receptors, guanylyl cyclase receptors, and tyrosine kinase receptors. Each type uses different chemistry, but in all cases, binding changes the receptor's activity and kicks off signaling. They play a role in apoptosis, cell differentiation, cell division, immune response, inflammation, just to name a few. But today, for my talk, I'm going to be mostly talking about RTKs. Now, RTKs are a major class of cell surface receptors. There are 58 RTKs in the human genome, grouped into about 20 subfamilies. If you look at the diagram in the bottom left, I will work through how this works, RTKs. But after ligand binding, RTKs form dimers that activate the tyrosine kinase residues. Once activate, these kinase phosphorylate themselves and bind other cellular proteins, and then go to phosphorylate other cellular proteins. The phosphorylated cellular proteins activate a variety of signaling cascades. On the right are some RTKs you might recognize from either research, maybe in the news or anything, but these include your insulin receptors, PDGFR, FGFR, and so on. These are common drug targets in oncology and cardiovascular medicine. If you notice in terms of the extracellular structure, there's some variability, but in the intracellular structure, they share the same tyrosine kinase domain. This can be helpful in models where you can apply the same principles for each, we're only changing a few of the parameters. So why are RTKs important? RTKs matter because they can control cell proliferation, survival, migration, and differentiation. They are crucial so much that the dysregulation of RTKs is a hallmark of cancer, as it may lead to uncontrolled growth, angiogenesis, invasion, and metastasis. RTKs are also involved in resistance to radiation and in how tumors evade the immune system. That's why there are so many RTK targeting drugs. And from a modeling perspective, RTKs offer a rich playground. There's complex biology, lots of data, and clear clinical relevance. This table is a list of dysregulated RTKs and their occurrences in some pretty serious tumor types, but you have the RTKs and the gene associated with them, and then the tumor occurrences and where they're related to. So honing in on angiogenesis or neovascularization, this is the growth of new blood vessels from pre-existing vasculature. It's essential in normal processes like development, exercise, wound healing, and the female reproductive cycle. But it's also involved in over 70 diseases, including cardiovascular disease, breast cancer, and diabetic retinopathy. Because blood vessels are so crucial, signaling pathways that regulate angiogenesis, like VEGF and VEGFR, and just to define those terms, that's vascular endothelial growth factor and its receptor, have become major therapeutic and modeling targets. So tumor growth and metastasis requires VEGF. I'll be showing a few pictures from Genentech showing the vascularization of a tumor. You start with an avascular tumor, and usually this type of tumor can grow up to about 1 millimeter cubed without vascularization. And when the tumor reaches that volume, diffusion alone isn't sufficient enough to bring oxygen to the cells. This triggers the secretion of VEGF from the tumor, and then VEGF then binds to receptors lining the nearby blood vessels. This triggers a signaling cascade, which in turn initiates the growth of blood vessels towards the tumor. And once the tumor gets vascularized, the prognosis of the patient becomes very poor since now the tumor cells can metastasize and invade distant sites. This diagram shows the complexity of VEGF and VEGFR interactions. Multiple ligands like VEGF-A, B, C, and D, as well as PDGF, which is placental growth factor, interact with different VEGF receptors in different isoforms. It's not a simple one ligand, one receptor story. That complexity is exactly why systems biology approaches are so useful in this context. And instead of guessing, we can actually build models that capture cross-reactivity and predict how shifting one component affects the whole network. Now we connect this biology to modeling. So quantitative biology, this uses biosensors and experiments to measure things like ligand concentrations and receptors levels, as well as signaling activities in the vascular microenvironment. Then there is computational biology that then takes those measurements and builds mathematical models and simulations. When we merge the two, we can both fit models to the data and use models to generate new testable hypotheses about signaling behavior. Now, what is a model? Model is an abstract representation of a real system. In our context, it could be a simple diagram of a biochemical network or a system of differential equations that describe how concentrations change over time. Models help us and explain and predict behavior. When we write down a model for a signaling pathway, we're making our assumptions explicit and giving ourselves a tool to simulate what-if scenarios. So here in the bottom left is, I guess, biochemical reaction, very simplified, but the dots represent the metabolites and the arrows represent the reactions. So C produces D and A produces B. What the line coming from D stands for is that it's an inhibitor for A and B. A very simple biochemical reaction model. Modeling is basically what helps us digest complex signaling pathways like this. Now, there's a quote by George Box, a British statistician, who said, "All models are wrong, but some are useful." The goal is not a perfect replica of reality, but instead a model that's good enough to answer specific questions or guide experiments. Modeling is subjective and selective. We can't include everything, so what's important for the question, so we decide what's important for the question. For example, Michelin-Menten kinetics work well for many enzymes, but not all. On the right, it's a curve which expresses the relationship between substrate concentration and initial velocity. And this has a shape for most enzymes, a rectangular hyperbola, whose algebraic expression is given by the Michelin-Menten equation. The model assignment is also not unique. The same biological process can be modeled in multiple ways. Different experimental methods give different perspectives and different mathematical frameworks, like ODE, stochastic models, or agent-based models. They all highlight different aspects. The choice really depends on your problem, purpose, and what data you have. Multiple models can provide complementary insights, but they can also be hard to reconcile. So it's important to be clear about your assumptions. Now, computational modeling also has several advantages. It forces you to clarify concepts more so drives you to clarify concepts and identify gaps in knowledge. You can sort of stretch time and space. So let's say you have 12-hour assay. Instead of having to wait after all 12 hours, you can just model it. And if the model is good enough, you'll get the kind of results that you're expecting. You can also explore many conditions pretty quickly. Models don't harm patients. They can help reduce animal use and focus lab experiments on the most promising conditions. And ultimately, a good model helps you make testable predictions. Now, focusing more on targeting angiogenesis, as was said, VEGFRs regulate angiogenesis and is pretty pivotal in diseases. Now, anti-VEGF therapeutics do exist, but they do face some resistance, and this is because the story is more than VEGF and VEGFR. It actually involves other ligands like PDGF or platelet-derived growth factor, which also bind to VEGFRs. And this kind of cross-family binding is underexplored. So research wants to look at novel interactions using computational modeling. And the reason why it's trending more towards computational modeling is because experiments are just too timely and expensive. When it comes to identifying novel ligand receptor binding partners across protein families, we want to look at what exists and what is plausible. So again, prior work has revealed that PDGFs can bind and activate VEGFRs, and this has some functional components, affects endothelial behavior and tissue permeability. When we take a look at structure again, we see some similarities in these families that we focused on thus far. If you look at PDGFR and VEGFR, as well as FGFR in the schematic right here, there are also other families that include this as well, but that sort of cusp, the reverse C that represents an Ig-like domain, these are shared in a lot of these RTK families. And then also for the ligands, there are also another shared motif called cysteine knots. We suspect that these may play a part in the crosstalk between these interacting RTK families. So we've talked about computational modeling, but if you keep up current with the news, it's hard to avoid the recent boom and push for AI. So what is AI? In very simple terms, it's a program that can sense, reason, act, and adapt. Now, machine learning or ML is a subset that enables computers to learn from data. Instead of being explicitly programmed for every scenario, algorithms are designed to automatically identify patterns, acquire knowledge, and build rules for large data sets, mimicking human intelligence to achieve specific goals. This learning process allows the model to improve their performance on tasks as they're exposed to more data. Now, DL, or deep learning, is a subset of machine learning that uses neural networks with many layers. So in the bottom right is a pretty basic neural network that you see. You have an input layer, you have a few hidden layers, and then you have your output layer. Now, it can handle complex nonlinear relationships and diverse inputs like sequences, 3D structures, and network data. With this, we're able to process diverse biological data, in our case, to measure interaction possibility. And again, this data includes protein sequence, structure, domain interactions, and gene expression. These stages hold true for a lot of applications, but again, for our context of protein-protein interaction prediction specifically, first, we want to gather data and label known interacting and non-interacting pairs. Then we want to label these, extract features, train a model, evaluate the performance, and finally deploy it to predict new interactions. To emphasize, researchers want to map these PPI networks to understand disease mechanisms and discover new drug targets. AI PPI prediction involves the use of structural and sequential methods. So this is sort of a timeline of what's been done so far. What you see on the left is machine learning models, the legend of it. You see the DNNs, CNNs, GCNs, these are more sort of like the neural networks. Then you have more conventional machine learning models like the support vector machine, the SVM. And if you can see and notice, as it's trended more current, there's more deep learning approaches that have been researched and used. Respectively, in terms of structural and sequential methods, their application is limited by availability of experimentally determined structures and challenges in capturing complex nuances. Researchers seek insight towards the computational boundaries and strengths of different model types. I am researchers, and evaluating PPIs through various classifier combinations, I feel, can help better uncover interacting proteins. Our work in the lab has looked at comparing different machine learning classifiers, including our own model, which is defined as a sequence-based PPI classifier using composition-based sequence encoding method and a Genasi pooling pair encoding approach for classifications. A whole lot of words. But we have already done this, and I've added my own research into it by researching some more current methods. So again, this is the neural networks and everything. And the conclusion I came up with, first I produced this graph, then I also came to the conclusion that graphical classifiers using sequential and structural data result more so in greater accuracy, as well as F-score. And then F-score is a metric that combines precision and recall. It's a single score that indicates the overall effectiveness of a classifier. But with this, I've used this insight and then also explored individual models as well. So this is string. The dots or nodes are the proton groups, and the edges are the predicted interactions. So string is a database that basically gives you this. You input a protein group of interest, then it uses known databases of interacting proteins and non-interacting proteins, as well as some predicted interaction from stuff like co-occurrence and I think fission fusion methods. But it comes up with this. In my head, when I first started doing with this research, this is kind of what I wanted to produce, sort of like an interactome of RTK and cross-family interactions. But wanted to combine everything, and I proposed an approach of a unified platform for standardized model benchmarking and clear performance comparison. And here I landed on PPI Insight, which is a tool for automating protein interaction benchmarking. So this is my accelerator project. My designated eScience data scientist, again, was Dr. Dave Beck, and through CSE 583, a course in software development for data scientists. I also work with another person from the eScience Institute, Dr. Bina Hazelton, and then my classmates, Maya, Rita, Fiona, and Walter. So there's no conceptual framework involved assembling positive and negative interacting data, using Python to create a structured database, surveying the available protein-protein interaction predicting models, writing a wrapper functions for this model to standardize the runs, doing this for multiple models, then ending up with cross-family interaction data for the RTK sphere. That has translated into this. So split up into 3 tasks, mostly for development. but it starts off, a user inputs search terms for proteins of interest into our tool, which then scours a lot of protein databases. What we use or settled with is Unipro KB, and it outputs FASTA and or PDB files containing sequential and structural protein data. This is then stored somewhere. In terms of our repo, it's stored in our repo. And this data is then used as input for a collection of protein-protein interaction prediction models. These are then run, they dock a pair of proteins. So basically they see which conformation is most plausible, and it output metrics that assess their likeness to interact. And then these metrics are stored into a spreadsheet and then plotted on a graph and uses to compare the interaction scores for a specific PPI pair across the different models that we use. And this graph mimics the graph I showed earlier about all the classifiers on the x-axis and then whatever metrics you want to see on the y-axis in terms of one that determines how likely it is to interact. Again, the tool uses the UniProt API to access the data. And the UniProt is a protein sequence and function info database. This is a good option because all the proteins in the database are reviewed by the Swiss Institute of Bioinformatics, which ensures that all data are high quality. The API also lets us use any program language to interface with the database website. So it's a good option for users who can come from diverse programming backgrounds. In terms of the computational models that have currently been implemented, we have HADOC, which stands for High Ambiguity Driven Protein-Protein Docking. It's a widely used computational tool for the integrated modeling of biomedical interactions. Then there is LightDock, which is a framework for predicting how proteins, peptides, and DNA dock together. But of course, we're focusing on proteins. Then there is PyRosetta, or more specifically Rosetta Doc, which is a Python-based interface to the Rosetta molecular modeling software. It allows for custom scripts for tasks like protein-protein docking, which is perfect for us. So who would use this, you may ask? Let's say a graduate student named Anna is investigating whether two proteins interact under different cell stress conditions. She wants to quickly fetch sequence and structured data, run several PPI models, and compare their outputs in one place. Anna wants a workflow that reduces the amount of steps for this process. She is comfortable with coding in Python. Now, just curious, by show of hands, does anybody relate to Anna at all? Okay, we got 2 people. Means I'm doing something good. In this video, I'm just going to go through the repo page and also look at the structure of everything. Most of this was structured for CSA 8583. So if any of you have taken the class or are interested in taking the class, it's very helpful. It was a major help for actually knowing how to structure everything and not only kickstart it, but have a pretty much good developmental phase. So the tasks are ran through the Python scripts. When the data is created, it's stored in their own directories. I'm just going through those for those who are familiar with the structure of these files. These are like PDB files. PDB files, basically, they give coordinates for each atom in the protein. There are also example inputs and outputs to show what the process is expected to look like. Again, this is the PDB files that are showing. It's a pretty long video, so there are also scoring metrics that that are given as a result of running these models. It's shown later in the video, but this is also going to be shown in the other slides, so I'll talk about this a little bit later. But first, I wanted to kind of divide this up by stages. So in stage 1, again, this is mostly for task 1. Starts off with fetching protein data. What do? You search for the proteins you want, as you would on the database website, and it returns queries and data files, and this is stored in its own directory. Again, if this is familiar, this is the PDB file that I was showing. Here, I'm going to show what it looks like to run one of the models. So again, there's HADOC, LightDoc, and Rosetta Doc. Right here, I'm going to show LightDoc. So you call the script from the command line interface and also reference the data files you want to use. You see, just by calling the script, you get some help for the usage. So again, I'm going to call PDB select doc, which calls the light doc model, and then I'm going to use these input files. These, again, are examples already stored in the repo. I think I sped this up by about 20 times, so it definitely isn't this short of a process. I wish it was, but there's active parameters that users can change. In the command line interface, you can actually alter the run to allocate more computational resources to get these results. Now, after you get this, here's an example of a visualizer. So the metrics are stored in spreadsheet files and are then plotted with the model on the x-axis and the metric of interest on the y-axis. So again, what I showed when I had the machine learning classifiers on the x-axis and stuff like accuracy and S-score on the y-axis, but this could also include the RMSD, doc score, really just anything the user may be interested in. So predictions from these models aren't the end of the story, they're just hypotheses. For my research, I plan to validate predicted interactions with experimental PPI assays, and these validated results will then feed back into functional and conformational studies and into updated computational models. This creates sort of a loop where computation and experimental data refine each other and ultimately help us understand and modulate angiogenesis more effectively. In terms of the accelerator project and what my classmates and I learned from working on it for CSA 583, this was my first time using GitHub, and it's a really great way to streamline communication. It's also not my first time doing project design, but first time doing project design for a computational project. It helped trying to figure out how to delegate certain tasks to certain people. So we basically match people by their strengths and weaknesses. And all in all, it came out pretty well. So now I just wanted to acknowledge the Department of Bioengineering, East Science Institute, the grants that I work on, of course, the research lab with Dr. Princess Mokade, and everybody in it. And then I wanted to give a special thanks to Drs. Dave Beck and Brina Hazelton, as well as my CSE 583 classmates, Fiona, Maya, Rita, and Walter. And then also thank y'all for your time and listening. I'm also open to any ideas or suggestions or any questions that you may have. I'm going to open up, of course, the floor to questions, but let's say you leave and then you think of a question, please feel free to contact me at keku@uw.edu. That's my last name at uw.edu.
Right.
Yeah, so some of these models, okay, so just to make sure I heard you right, you asked how many predictions are output as a result of running these models? So for some of these models, I know for LightDoc, there's clustering methods that take sort of the best predictions, and we plan to take the best predictions and take an average of that and then plot it, if that answers your question.
Can I follow up on that? An average structurally or an average of the document free energy?
An average of the document free energy estimate, because the structure, it's kind of hard to average this. You would have multiple structures, but in terms of the actual score, which is the quantitative value, this would be averaged. And when the graph is produced, you have an average value for that specific protein-protein pair and that run.
Maybe follow up on that. So the average prediction is the probability of the interaction, positive interactions that we find the model.
Sorry, can you repeat your question?
So you get an average prediction based on the models, you go through the interaction models.
Mm-hmm.
And what is this an average of Like what value is it possible to graph?
Yes. So I'll see if I can go back to I'll see if this video runs fast enough. But when LightDoc runs, for this case, it produces about 100 folders, 100 conformations, and each of those have about 10, at least due to these default parameters, 10 scores that they have. And when you cluster these, they of course pick the top 10, but if you don't, you would have to go through all of these and pick the best ones. So it's not going to have one prediction after the run. It's going to have multiple because again, there's many different conformations for how two proteins can actually dock together. So it has to go through all of these and give you scores for that. And when you have those scores, at least for what I'm doing and for what this tool is doing, we're picking the top of those scores and averaging together for the graph. Does that answer your question? Yeah. Okay. You said, so to repeat the question, you said I showed a slide where sorry, can you repeat again? This right here? Oh, the graph right here. Yeah. Yeah, so we have a table of interacting RTK cross-family binding data and non-interacting data. And then we also have a standard interactome data set. So all of these classifiers were trained on the standard interactome data set and were then tested on the interacting and non-interacting pairs. And as a result, we got these values for accuracy and F-score.
Other questions, right?
Just curious, what were the challenges from computational side of things to get this up and working?
Mhm.
Apart from the ramp-up time of getting into the zone, but challenges with getting the code ready or just too much to compute and how do you work around all of those constraints?
Yeah, I think the biggest and maybe most frustrating issue was trying to integrate Haddock into the pipeline, and I actually got some or try to get some help with Dr. Beck with this. I think it was something to do with, I think it was like the CNS architecture of Haddock that Haddock wanted to use. Yeah. So I had to get Docker images for some of the models to work and basically run these on Hayek as well, which is a supercomputing cluster. So there's some issues in that, but of course they're overcome. But I'd say that's the biggest issue, just actually taking the models and integrating into the tool. And I think that will be the biggest issue moving forward is seeing which tools actually fit into our tool, which models will actually fit into our tool.
You want to say just a bit more about what Hayek is?
Yeah. Hayek is UW's supercomputing cluster. So it was very helpful for me because instead of running it locally, and let's say, I mean, you see my Mac is from like 2019, it runs off Intel. I think it has like 8 cores or something and like 12 gigabytes of RAM, but when you're running through a supercomputer cluster, you have a lot more RAM and a lot more computing power. So Hayek is your go-to for that. It's a cloud-based system. Yeah, if you have anything computation you want to run, I recommend using it or looking into it at least.
Again, a curiosity question. I'm not from biomedical, so maybe a naive question. So the step one was to fetch protein data, and what does their interaction look like? Is it that I want blah protein and this Y protein and get me PDGFs from them? Is that how it works?
Yeah, pretty much. So the reason why I wanted to integrate it into this tool was because before the tool, what I would do is I'd have to find the database that I wanted to use. Then I'd have to see which protein that I actually wanted to use, type it in the database, filter through the queries, and then download the PDB or FASTA files. With this part of the tool, task 1, what it does is now you have either through the command line interface or Jupyter notebook, you just type in the name or ID of the protein you're interested in, and then it will give you some queries and then also automatically download the data for you. So it just streamlines the process.
Great talk. I'm wondering if you have a chance to actually use this tool to drive some interesting insights to VEGF cross reactivity.
Yeah, I haven't used it yet, but with the way it is right now, I could. But I know how much further I could take it, so I haven't actually input it into my research. I will say though, I have used similar models for cross-family ligand interaction predictions. And what I didn't include here in terms of, let's see if I can go back here, cross-family binding. Of course, again, I talked about PDGF and VEGF. I also found out that the FGF family also interacts with the VEGF family, the VEGFR family. And again, that's just through research and then also using his models. So stuff like that is more so why I created the tool and what I hope other researchers can use it for. Yep.